So thank you. I really enjoyed the meeting this morning, and I hope we'll be to do a great job as well with Andreas. This Congress was the opportunity for companies to introduce our new alliance between Oncodesign and MBX0, as we have highly complementary expertise. Oncodesign in preclinical development and MBX in clinical development. So we try to give you some insight of both our expertise. So myself, I'm the head of a pharmacoimaging group at Oncodesign and targeted radiotherapy. And Andreas, you want to introduce yourself?
I'm Andreas Kluger. I'm the general manager of MBX0, a radiopharmaceutical addicted company for 20 years. And yeah, we are very pleased to have now this alliance with Oncodesign, which basically combines the world of real-world clinical development with the complete extensive world of tumor models and other disease models, which you find hardly anyone surpassing Oncodesign. So we think Oncodesign, specializing also in radiopharmaceutical development, we are a nice fit. So thanks, Cyril. Go ahead.
Thank you. So it takes two to tango. Yes, that's how we want to see it. We heard it this morning. We need many expertise in these fields, and so this is one way. So I'm going to try to move the slide. Yes, first slide. So starting targeted radiotherapy starts from radiochemistry and bioconjugation to first in man. So there are different expertise you need to go through. Of course, the radiochemistry and bioconjugation, the in vitro binding assessments using different biological samples, the biodistribution and the dosimetry. We heard a lot this morning. And of course, you need to do your proof of concept in animal models and evaluate the toxicity before you can enter in phase 1. You have many isotopes you can work with. Here we highlight 3 of them, just because there are three types of isotope: the lutetium beta, the iodine-131, or actinium-225. And besides this therapeutic isotope, usually we always develop an imaging companion diagnostics. So there are many options. We heard of it also. It can be fluorine if it's not metals, but otherwise it could be technetium, copper-64, indium-111, gallium, or zirconium. And to choose the right one is not that easy. And you need to understand the logistics, the availability, the half-life, and so on. So we try to give you some insight here for that. So maybe if you want to address some points in the clinical trial, and I will give the hands to Andreas to really talk about it. So, what needs to be addressed in clinical trials, and in the next step to decide what to be done at the preclinical stage. Andreas?
Thanks, Cyril. So basically, we have the 3 classical stages of drug development, phase 1, 2, 3, which is known from the non-radiopharmaceutical world. And in analogy, but slightly different, we also have it in the development of predominantly radiopharmaceutical therapeutics, which are more special than diagnostic, let's say. We have typically to identify the maximum tolerated dose, which is a double dose, which is at one end a radiopharmaceutical activity dose, right? So gigabecquerel or curies. And at the other end, we have also a mass dose, which also is of importance in the development of radiopharmaceuticals. And since this is the mode of action of radiopharmaceuticals is multiple, we have also to optimize for the dosing intervals. It's not obvious from the beginning that we dose weekly, daily, or monthly, or even more monthly, with more monthly intervals. So that is what we have to characterize. That's the administration mode, how to achieve the best result. Of course, another aspect is the characterization of safety, which is a function of this administration mode. Of course, you can imagine that if you dose, for instance, a therapeutic in weekly intervals, you have a different bone marrow toxicity, for instance, if you compare to a situation where you dose it in monthly or 3 monthly intervals. And tolerability is a sort of softer form of safety, which usually is addressed by recording health-related quality of life data. Then you have the third step is, of course, the most important one, which factors in from a pharmaceutical perspective into the value of the asset. So does the drug work? First, proof of concept, which is typically addressed in the setting of in the setting of a phase 1 study already. We look for the right dose, which achieves the best effect while minimizing the side effects. And in later stage trials, phase 3 trial, we then want to confirm the efficacy using confirmatory statistics. So basically, the complexity, it has already been mentioned before by Mauricio Mariani, but I'm going to repeat it here. It's really the logistics of the pharmaceutical production, which have to match basically trial recruitment, because you can very easily frustrate patients and also investigators by not being able to produce and bring your drug to the patient at the time when the treatment is planned. We have the aspects of specific activity, which I have introduced already. We have a mass dose and an activity dose to optimize, and we do that basically by measuring dosimetry. So these are specific aspects of radiopharmaceutical developments: dosimetry, specific activity selection, and production logistics, which mainly differentiate a radiopharmaceutical development from a conventional drug development. Next one. So here again, the 3 phases of drug development. What are we looking for in phase 1? We want to generate the basic information about our drug, which is the biodistribution. And in case of radiopharmaceuticals, the dosimetry question, which is based on biodistribution imaging. We want to see whether our drug actually targets the disease, typically a tumor disease, but increasingly, as we have heard from Chris Bernbruch, also other diseases such as rheumatological diseases are potentially targets for radiopharmaceutical treatments. We want to identify the maximum tolerated dose, as in any drug development. As alluded to previously, this is mostly the activity dose we are looking at, and not the mass dose. We look for safety and PK and metabolism, and of course, first evidence of activity, because most trials are phase 1 trials run in diseased populations already. So we would, of course, like to convey an individual benefit and look early on for whether the drug at the treatment concept is flying. Phase 2 is the trial phase, or the development phase, where we actually de-risk our development, where we look for the effect size, how much benefit can we actually achieve using this treatment, in order to then plan a confirmatory trial for the later phase 3, which is then the basis for the marketing authorization. So now please put the flash down. It's not yet. Yeah, the holistic design is the summary. So we want, in the context of phase 2, we want to identify different patient subgroups which particularly benefit from the treatment. For instance, patients with first, second line situation, high risk, low risk, patients of a particular disease group, in order to identify the best patient population for our later stage confirmatory phase 3 trial, in order to make our drug look good in front of the regulators, because that's what we're aiming at, or you as developers of radiopharmaceuticals, to have a drug which has a nice safety profile and a nice efficacy profile, because although that should be not done, everybody does cross-trial comparisons in order to, for instance, compare drug A versus drug B, which have never been compared directly in a randomized compared trial, but it's done. So you should basically select the right patients where you have a good safety and efficacy profile in order to look also in the inter-product comparison, look nice in the interest of your asset. And another phase 2 question is typically dose finding in conventional drugs, where you, for instance, would compare placebo versus dose 1 versus dose 2. Since radiopharmaceuticals have a special mode of action, which is actually multimodal, the dosing interval may also factor in differential efficacy. We have seen this morning, for those who have attended, that basically the immune system plays a role, and the spacing of the different, mostly repetitive treatment administrations can be of importance for maximizing the treatment effect. So rather than comparing a 20 versus 40 milligram of a drug, we would rather compare a treatment interval of 4 versus 8 weeks, for instance. This is also a phase 2 question in radiopharmaceutical development. Phase 3, we then, of course, do a comparison to standard therapy in a confirmatory statistically design in order to get our marketing authorization using typically randomized controlled trial. So the holistic approach basically means that we think, when designing the phase 1 study, how to maximize early on all relevant information about the drug for later stage and for a swift development. Often phase 1 trials are designed basically not in order to address a maximum of questions which we should tick off from our development plan, but in order to quickly acquire additional data, evidence for investors, which is a problem of our field, but it's getting better to the success stories many of the participants of this meeting here have produced themselves. So funding for a decent and sufficiently powered phase 1 study is increasingly less of a problem than compared to earlier years. Next one.
So if we go back to the hear me twice. If you go back to initial steps of the radiopharmaceuticals, we should think properly about the radioligand design, and we discussed earlier this morning if we should have 1 compound, 2 compounds, what will be the ligand and radionuclide pairing. Should we consider the PK of the ligands and the half-life of the nuclide? Could be related. Chris said that might not be the key issue, the half-life. But of course, if you want to use gallium-68, for instance, with antibody doesn't make real sense because the PK of the antibody will not match with gallium-68. So this has to be taken into consideration. And of course, you need to consider the chelate agents and how you will do your chelation. If you need to use heating, if you need to use some pH, depending on your pharmacophore, you might be able to heat it, or to use acid pH, or you may not be able to use it. So this is of course something that you need to consider. The chemistry and the radiochemistry of zirconium, for instance, or for lutetium will not be the same. So these chemistry conditions are also of importance related to your pharmacophore. And of course, specific activity is crucial. It's easily managed at the preclinical settings, but you need to consider what will be the specific activity you can achieve at the GMP level and evaluate that eventually immediately at the preclinical level. It doesn't make sense to use huge specific activity if it's not reachable at the GMP level. And considering the bioconjugation itself, you need to consider the number of chelators you want per molecules. It's not the same if you have a small molecules or if you have an antibody, if you can use a random or site-specific bioconjugation. And if you want to introduce a spacer between your chelator and pharmacophore, like for PSMA with a BSA binding domain, to modify your PK. So all this consideration Now, we have a lot of knowledge about it, but we are clearly in the field of drug optimization and not just testing one compounds after another one, but we can really think about the drug optimization process. Here I just highlight some pharmacophore and imaging radiometals isotope because also pairing, and you can consider SPECT or PET. This is also something to have in mind. So on the next slide, we want to highlight the radioligand production. As I say, validation, the radiochemistry at the preclinical level, usually, so I will not say it's straightforward, but we have tools to develop it quite easily. The basic quality control is done usually with ITLC or HPLC, and we do the different validation, especially for the stability at different conditions, storage condition. And one of the points that we want to address is usually the radiolysis. This step is not very difficult, but you have to keep in mind what is necessary to be done at the GMP level. And maybe Andreas, you want to highlight that.
Yeah, thanks. For the development of new radiopharmaceuticals and basically designing and placing the trials, you should have early on a concept where about you want to go with your development project, because clearly, especially with short-lived radionuclides like gallium and fluorine, you typically would like to have your manufacturing of your product on the same continent at least, or ideally in the vicinity. Typically, we work today with multicentric study settings in order to expedite recruitment, as is the common standard also in the development of non-radiopharmaceuticals. So here, basically, the standard solution is to have a single site developing the technology for a synthesizer-based automated GMP production of a radiopharmaceutical. And if due to the half-life of gallium and fluorine in the, here to be mentioned as short-lived nuclides, the distribution is not possible to all trial sites, you would clone such a GMP production facility and have them distributed, for instance, one in North America, one in Europe, and one in Australia. Now, that's a typical setting for global trials requiring production of a uniform GMP radiopharmaceutical. So logistics, including customs clearance, and transport solutions is something which is vital to the successful running of especially later stage radiopharmaceutical trials, which is what a CRO would provide as a service. So next one.
So then when you move forward in the development, you have some in vitro validation to perform. And the first one is to check the affinity of your compound. Adding chelating agents might impair your affinity. So you have different assays. You can evaluate, the immunoreactivity. You can use saturation or competition assay. And this is straightforward assay that has been done immediately after the bioconjugation and the radiolabeling. Sometimes isotope might also modify the link to your targets. Then you can evaluate if your compounds will have an intracellular localization, depending on the isotope. It might have some difference, and the cytotoxicity of your compounds. What is the difference between the compound alone versus the compound with the isotope, to evaluate clearly the impact in vitro before going to in vivo. This is what is done typically at early stage, and when we move to IMPD file, there are some more deep questions that need to be analyzed, and maybe Andreas, you want to highlight what is needed for IMPD file.
Well, essentially, IMPD is consisting of the CMC part, which is essentially the quality of manufacturing for the clinical trial material. And the biological part of the IMPD covers essentially what you just mentioned, the data generated on in vitro binding, on in vitro stability, on plasma protein binding, and any preclinical in vivo data as far as available, so as to convince our investigators and of course ethics committees and regulators that it's ethical to start a sufficiently characterized compound in a first in human trial. That's the prerequisite. So we need the preclinical data package in order to start a first in human trial. And that's basically also the particular charm of the Oncodesign ABX0 collaboration. We basically know what we need for first in man. We have a dedicated toxicological and pharmacological expertise group, which allows us to basically minimize the general toxicological burden and at the same time provide best in class, as we believe, preclinical pharmacology data. You have to appreciate that the guidelines for toxicology are different for oncological products, for radiopharmaceutical products, and for biological. So you can essentially not follow the guidelines. You have to have a pharmacologist who designs an individual program for an individual drug candidate, depending on the pharmacophore, depending on the characteristics of the nuclides and so forth, which is part of our joint service offering. Next.
Yeah, and this is one of the key aspects, as you say, the biodistribution and the pharmacokinetics at the preclinical level. We go straight to in vivo biodistribution to evaluate specific updates, percentage of uptake in the tumor or the tissue of interest with the in vivo clearance. Do you go for kidney, for liver? What are the ratio between the two? This is clearly essential. And to determine the time of activity curves and all your basic data set for dosimetry. And dosimetry, this is one of the best strengths of ABX0 with their software Kudos. And I see George is popping up, so we are running out of time.
We can give you another couple of minutes if we've accounted for the slightly slow start. So if you just wrap up in the next couple of minutes, that'd be fantastic.
Okay.
Yeah, thanks. So Andreas, do you want to say something on Kudos and shortly?
Well, basically, the charm is that we will translate based on the preclinical biodistribution data. We will forecast the human dosimetry, which is part of the IMPD package for any radiopharmaceutical. That's a service provided by the Oncodesign and ABX0. Once we are then actually in the clinic, we have to run dosimetry and verify in how far our preclinical predictions are actually valid. So we identify in dosimetry, it's not a burden. It's really something to learn how to rationally use our radiopharmaceutical compounds. We identified the most exposed organs. Typically, we look for sensitive organs like bone marrow, kidneys, and also the lungs. We determine the individually absorbed dose to the organs in terms of milligray, that's the absorbed energy dose per megabecquerel, to have a uniform normalized value to also allow comparison between different radiopharmaceuticals. And we determine the effective dose to the whole body, that's in millisievert of megabecquerel, which in addition to the milligray also contains a biological weighing factor of how much you get insensible as opposed to less radiation sensitive organs. That's what we call safety dosimetry. But what's more important, of course, if you think of drug development, you have safety and efficacy. The efficacy dosimetry is that what we actually get to our tumor if it's an oncological product. And this is also in milligray, and it is, of course, the same unit as you know it from external field radiation therapy. And we are in an interesting time starting to understand what particular gray dose conveyed by our radiopharmaceutical is needed in order to actually control tumor growth. So all that can be determined using the Kudos software. which is similar to Olinda, but has additional features. Yeah, next one.
Yes, so beyond dosimetry, we have to evaluate the efficacy, and here this is where Oncodesign is, I think, is very strong at, the choice of model, and of course it has to be not to be radiosensitive. So if you can avoid SCID background, for instance, this is something to pay attention, because if you have already some toxicity related to the mouse background. To talk about the species cross-reactivity, if you need to use syngenic, xenogenic, or humanized mouse models, that's something that is even important to consider. Of course, that recapitulates your pathology or indication. And if you have data on these models, when you want to start a combination, if you have pharmacological data with standard of care, this is also something crucial when you want to move to later stage. There are different parameters to be addressed. The dose, we usually try to work with a range starting at 15 up to 120, even if it's very high. Single dose escalation, what kind of schedule and intervals you want to use, and as Andreas say, the specific activity, the mass ratio. This is really something that is very different than regular drugs, and has to be really to understand how this is working. And the final readout, here you have a nice complete response with a compound related to lutetium. And I hope that all compound we test for our different clients will look like this one. And then, we have a full package with tumor response, imaging, biodistribution, and dosimetry, and biomarker target expression. I think for the time being, we have to skip a little bit the toxicity, as Andreas have already mentioned it. One of the key things is you have to determine the toxicological program either as a microdose, if extended single dose toxicity for diagnostics, or for therapeutics that would be individualized design depending on your pharmacophore. And we can come back to this point later on if you have interest for the toxicity design. And one thing that we wanted really to highlight, because this came back many times this morning, is the combination study. And we consider two types of combination, with the one with chemotherapy or the standard of care, when you have to evaluate the potential synergies. And there are different steps to address is the MTD of your chemo agents, the dose efficacy, the MTD combination, and clearly to evaluate the combination at the different efficacy, PK, and safety biomarkers. So finding the dose for combination is not that easy in mouse models, but this is something that we have good expertise with based on our long-term knowledge of standard of care. In combination with immune checkpoint can be even more challenging, depending if you want to evaluate the abscopal effect, if you want to evaluate the immune infiltrates. Here, as this cartoon may illustrate, that radiotherapy might convert a cold tumor to a hot immune tumor. That can be addressed by flow cytometry or immunochemistry. We can work on syngenic mice if your target is your vector is cross-reacting, but otherwise we have to work with humanized mouse models, which is another challenge. Eventually it's a graft, and yes, we're done, John. I just wanted to highlight this recent study that has been published by the team of Malo and that's highlight the combination with PD-1, with anti-PD-1 in melanoma, with the melanin targeting radiotherapy, been published very recently. And you can see here that the combination with lutetium, you have a synergistic effect on synergistic effect if you combine PD-1 with lutetium therapy, whereas if you do the same combination with actinium radiolabeling compounds, you have no synergy. So just to highlight that these mechanisms are not far from understood yet, and experimental models are crucial to have that, and I think we have the experience to evaluate that. So if we want to conclude, the take-home message we want to highlight with Andreas is that molecular radiotherapy, and we agreed with Chris that we should find a common word for everyone. So molecular MRT is probably one of the more global one that we can use, is the emerging fifth pillar of oncology and beyond. Combining pharmacotherapy, radiation therapy, and immune activation is now clearly a multimodal therapy. MRT could provide, an unparalleled efficacy at the cost of a stunning favorable safety and tolerability profile. This is really, we have, believer on that. And the diagnostic approach can be considered as best in class when we talk about precision medicine. And the development of molecular imaging and therapy agents require specific expertise through the entire drug development. I think you will all agree on that. And this is why Oncodesign and ABX, have signed this alliance to really put both our preclinical and clinic together, and having a global network of experts that can help you to manage efficiently your drug development and take the right decision at the right time and as early as possible. And just
I couldn't say it nicer, so thank you.
So Andreas, if you want to conclude on the last slide, I'll let you the conclusion word for you.
Well, yeah, as I say, I couldn't say it nicer. It's basically, we all believe here that we are creating a new type of medicine, which is will be the cross-sectional medical discipline of the next 20 to 30 years, I would say, being both diagnostic and a therapeutic discipline. We have the privilege here to pave jointly with all participants of this meeting the way for this new medicine, and I think it's a privilege for everybody being here. I tend to say nuclear medicine is the opera of the medicine, or the opera of the natural sciences, so everybody is welcome to contribute from his discipline, and we try to be holistic with Cyril and his group to really conduct the orchestra on behalf of our customers and anyone who is interested in therapeutic and diagnostic nuclear medicine. Thanks.
Thank you both for the presentation. Thank you both and thank you ABX and Oncodesign for partnering with us for this year's meeting. It's been fantastic working with you guys. I can't say enough. Please head over to the demo area and check out the materials and the videos, and I am sure Cyril and Andreas will be happy to field any further questions. We do have the panel discussion coming up after Lin Jingmu's talk, so I'm sure we can touch on any questions there. We've got some great communications going on in the chat box on the right hand side there, so I'm sure Cyril and Andreas are going to delve into those right now while we have Lin Jing. But thank you both. See you shortly. If you pop the leave button just right there, and we'll bring Lin Jing on.
Thank you.
Great, thanks. Bye, everybody.